What do malaria-transmitting mosquitoes, crimson-hued pupils, and intelligence quotient levels depend upon? The astounding answer is that all of these realistic scenarios are determined by a polymer scientists have dubbed deoxyribonucleic acid, or DNA. Everything that humans have accomplished, from the building of the Great Pyramid of Giza to the extraterrestrial satellites, can be traced back to this pair of intertwining antiparallel polymers of Brobdingnagian proportions, housed inside of our cells. As crucial and beneficial as DNA can be, its malicious aspect cannot, should not, and must not be ignored. Acidic addles, murderous mutations, and stigmatizing syndromes arise from the blatant blunders of non-sequential genes. For centuries, no mastermind has successfully attempted to muddle with the intricate compilation. But a few decades prior to this day, a discovery arose that has changed, is changing, and will continue to change the foundations of medicinal and biological studies. Although unfamiliar to the majority of the population's awareness, genetic engineering has the potential to both benefit and harm humanity monumentally. Deoxyribonucleic acid, DNA, structure and potential. Is that an arbitrarily selected geometric figure? Is that a vibrantly hued truncated icosahedron? Is that a uniformly revolving autonomous walkway? Contrary to the common misconceptions presented, the diagram below is the fragment of a model depicting a DNA helix. The nucleic acid DNA, although intricately crafted and helically compressed, is composed out of moderately understandable repeating units known as nitrogenous bases. Nitrogenous bases, or nucleobases, are biological compounds that contain the element nitrogen. Nitrogenous bases bond with 5 carbon sugars, called pentose, and form a monomer which we call nucleosides. Nucleosides bond with a phosphate group to form nucleotides, which are the repeating molecules that form the backbone of both deoxyribonucleic and ribonucleic acid. The 5 nitrogenous bases that construct nucleic acids are adenine, guanine, cytosine, thymine, and uracil. In DNA, adenine A, thymine T, cytosine C, and guanine G are the 4 bases that are present, while in RNA, adenine A, uracil U, cytosine C, and guanine G are the 4 bases that are present. Thymine and uracil are interchanged into 2 nucleic acids because of the presence and absence of a methyl group in the molecular ring. Nitrogenous bases can be classified into 2 groups, purines and pyrimidines. Purines are more complex in structure than pyrimidines and consist of adenine and guanine. The less complex pyrimidines consist of cytosine, thymine, and uracil. Purines bond with pyrimidines, and pyrimidines bond with purines due to the structure of hydrogen bonds. The interchange of the nitrogenous bases form the antiparallel helical structure of the biomolecule. DNA is housed in compact protein folds called chromosomes in the nuclei of our cells. Each cell has 23 pairs of chromosomes, 1 set from each parent. Each chromosome contains uncountable numbers of units of heredity called genes, which you inherit from your parents. The probability of inheritance for simple traits like hair color can be tabulated by using a tool we call Punnett squares. More complex traits require more complex tools. The chromosomes are supposed to contain absolutely no errors in their creation, due to the fact that they are the blueprints of our cells. The genes that are being encoded are responsible for absolutely everything, from our height, our might, to our will to fight. Unfortunately, when replicating these precious creations, blunders occur, which mutate the genetic sequence and thus alters our existence itself. The human genome, or common language. All organisms have a sequence of genes that makes them distinguishable from others. Humans are no exception to this phenomenon. The variations in the genome presented within different organisms depend on a great number of factors, ranging from the amount of protein production to the respiration rate of the organism's cells. For instance, the porcine circovirus type 1 contains 1,759 genome pairs, while the bacterium E. coli has 4,006,000 genome pairs composing it. In a nutshell, the more complex an organism is, the more genomes are housed within their cells. The unique sequence that makes a human is denominated as the human genome. The human genome contains 3 billion genome pairs. The genome is composed of 23 pairs of chromosomes, each tasked with encoding aspects of our existence. The X and Y chromosomes exemplify this. They determine our gender. Genetic mutations pertaining to the genome occurs when there is a discrepancy in the number of copies for one chromosome. Normally, there are 2 copies for each chromosome, but in some cases, there may be only 1 or 3 copies. The terminologies used to define the chromosomal disorders are monosomies and trisomies. Monosomies and trisomies can result in malignant physical and/or mental effects. Instances of these repercussions include Down syndrome and Turner syndrome. In addition to these ominous anomalies occurring, when replicating chromosomes during the process of mitosis and meiosis, blunders can occur. Instead of a T, there could be an AC base pair, for instance. These blunders result in terrifying aftermath, such as cancer. These mutations can also occur due to environmental effects, such as chronic exposure to ionizing radiation such as gamma rays. To add on to this, the majority of genetic mutations cannot be corrected due to the ingrained format of the newly constructed nucleotides and the foundations of our DNA itself. Until recently, there was only waiting for the end to come. By the night of human discovery, scientists have discovered a radical and revolutionary cheat code for gene engineering, called CRISPR. Clustered regularly interspaced short palindromic repeats, CRISPR, the naturally artificial tool. In contrast to the alleged beliefs of many, genetic engineering spread from natural causes and developed within one of the simplest organisms on the planet, bacteria. Despite causing other eukaryotic organisms such a great magnitude of trouble, bacteria are under siege themselves. Bacteriophages are a type of virus that hunt for bacteria only. Bacteriophages kill, on average, 40% of the population in the oceans every day. This makes them incredible tools to utilize when attempting to destroy bacteria, such as instances when antibiotics do not function upon the pathogens invading the body. However, bacteriophages also cooked up another frenzy in biology. Most bacteria succumb to the attacks of the bacteriophages easily. However, for some rare organisms, an enzyme called Cas9 is able to steal some of the bacteriophage's genes and incorporate them into the bacterium's DNA. The next time a bacteriophage attempts to attack the cell, the enzyme Cas9 is able to identify the genes in the parasite, and the bacterium decimates it. CRISPR is currently the fastest and easiest way to edit our genes, which is the reason why a lot of investors are starting to rivet their attentions onto it. Unfortunately, this also means that there is an exponential increase in the quantity of greedy and headstrong individuals who seek to squeeze money out of people. The evolution of intelligent life, how we appeared in the game. All organisms inhabiting Earth today originated from the same primeval ancestor. Throughout the course of around 4 giga-annums, life itself has evolved from primeval unicellular beings into the intellectual and intricate Homo sapiens today. The theory of natural selection states that advantageous traits such as tall height or spectacular night vision will eventually become the dominant trait in the species because the organism with those advantages will be most successful, which leads to the eventual evolution of the entire species. Life forms known as archaea were the first life forms inhabiting Earth. During the Archean Eon, archaea first blossomed, being constructed out of the simpler classifications of cells, known as prokaryotic cells. These unicellular entities had the ability to inhabit extreme environmental conditions, such as extreme acidity or extreme heat, and were therefore perfect for living in the ancient Earth's environment. Further along the way, bacteria evolved from archaea after the environmental conditions had calmed down enough. Bacteria and archaea alike began to hunt for nutrients in order to carry out life functions, such as reproduction and energy harvesting. Unfortunately for those beings, the cells that they consumed were not enough to last long. The cells needed another way to obtain energy, and there was one almost infinitely efficient option: light. The photons hitting Earth had almost limitless potential to utilize for cells, and some of the bacteria began to evolve, and were now able to photosynthesize. These photosynthesizing bacteria were called cyanobacteria, and they provided themselves with a sufficient quantity of energy for nearly no cost to themselves. Bacteria and archaea that did not have the ability to photosynthesize began to target cyanobacteria as their nutrients to maximize the deal. Life did not evolve for a long duration of time afterwards, until one eventful day that created the first eukaryotic cells. An archaean conceived a bacterium, and the bacterium did not die. The bacterium no longer had to fight for survival inside of the archaeon, and the archaeon, in turn, provided nutrients for the bacterium to convert into raw energy. The bacterium became the first mitochondrion to exist. This hybridized organism had exponentially increased the potential for success. In addition to this astounding prospect, chloroplasts were once separate cells as well. Cyanobacteria were consumed by eukaryotic cells that were outlined by cell walls, forming the first fungi and plant cells. This in turn enhanced the energy output a single cell could produce, and life became more complex. Multicellular life first appeared in the game around 3 billion years ago, after unicellular cyanobacteria-like organisms bonded together to compartmentalize life activities. This proved to be incredibly beneficial to the newly formed multicellular organisms, as the already efficient individual cells can now focus on one task at a time, while its partners could add on to the finished products of the original cell. Hence, tissues and organs could build up entire multicellular organisms. Multicellular organisms also evolved throughout the remaining 3 billion years of Earth's history. The individual cells of the organisms adapted to better suit their structures and needs, forming the 6 kingdoms of life around today. Through the phenomenon of natural selection, the organisms in each kingdom evolved into individual species. Hence, an uncountable number of species formed, each with their unique characteristics. Genetic engineering, however, risked undoing the entire storyline of evolution of organisms right to the beginning of the plot. The Spliced Bedlam, the horrors of heredity engineering. Genetic engineering allows one to insert or remove the genes in any organism as desired. Though this is undoubtedly a superb triumph, it poses unascertained consequences. Firstly, genetic engineering contains the potential to alter the human genome. Once genetic engineering graces upon humanity at a wider scale, all parents might aspire for their future child to become constructed to be absolutely exemplary, and hence might resort to genetic engineering as a solution. Tall height, broad shoulder blades, attractive appearance, high intelligence, and great strength are just some of the traits that can be inserted per our whim. Unfortunately, defying the laws of nature for our personal benefit does not obviate the consequences that we will all eventually have to befall. For starters, a single engineered baby will affect every human being in the future, both in terms of our physical bodies and our ethical beliefs. The first edited organism will pass off their DNA into their children, thereby inserting a completely radical set of commands into our gene pools and therefore having the potential to change us as a species. Additionally, enhanced chances of success in life expands the number of opportunities of triumph that an individual can acquire. Hence, more people are going to become entrepreneurs, seeking to clamp down the rivalry between the other genetically enhanced humans who aspire to expand their empires as well. Thus, humans will expand more into the environment to exploit the resources. Genetic engineering also suggests that the global population will exponentially increase. After all, why would you leave your child or children with fatal diseases when they can live for an elongated period of time? Consequently, fewer people are going to die in the duration that takes for more engineered babies to be born. The amount of ethics and human rights that we hold true will also decline as genetic engineering becomes more and more prominent. Currently, our ethical beliefs still hold true due to the fact that every individual is unique in their own way. The DNA housed in every one of our cells are sequenced such that no two people are identical. If we experimented upon an individual, however, and manufactured the human in such a way that he or she becomes flawless, more individuals will follow, therefore tipping the gravity of our opinions. In a matter of years, the entirety of the population will become a monotonous sea of perfectness. Our perturbation of experimenting on ourselves will diminish due to the reality that no one is different. Why fret over one human when you can always replace him or her with an almost identical one? Finally, genetic engineering our genetic sequence poses risk for our environment and our economy. Individuals will lot of the capable qualities of becoming triumphant entrepreneurs will aspire to become them, and hence set their sight upon the resources embedded into the earth, such as minerals or Arctic water. This heightened rate of success and interest in environmental resources results in a faster degradation of the conditions of nature itself, and thus the environment will deteriorate along with our ethical concerns and our biodiversity. The icing, the advantageous traits. Albeit containing blatant flaws, artificially constructing our genetic sequence is incredibly potent in destroying maladies and diseases. A clear instance of this in the real world is the course of action that we conduct against malaria. Malaria is a fatal tropical disease that is caused by microscopic parasites referred to as plasmodia. Plasmodia hitch a ride upon mosquitoes. When the infected mosquito bites down upon another organism, the plasmodia attack the organism's cells in order to reproduce, dragging the host organism's health down to fatal conditions. Current methods of combating malaria are proving to cause more damage than they patch up. For instance, in Borneo, the WHO utilized an insecticide called DDT to eradicate the mosquitoes that transmit malaria in the neighborhood. However, an unanticipated repercussion was that other organisms were also adversely affected. After a chain reaction, the WHO initiated Operation Cat Drop. Utilizing genetic engineering to crack the catastrophe proves to be a safer alternative. By using CRISPR, scientists are able to manipulate and alter the sequence of the alleles being passed down from the parent mosquitoes to the offspring. A concept of genetic engineering known as the gene drive can be utilized to alter the probability of the various types of alleles from occurring. The dominant genes of malaria-transmitting mosquitoes could be altered so that they are naturally inclined to not transmit the malignant traits that are attracted to plasmodia. The traits that are inclined to carrying the parasite are converted into recessive genes and inadvertently appear in the minority of the population. Hence, the number of malaria cases will decrease exponentially, and over time, the baleful genetic sequence will be smudged from existence. Furthermore, inevitable phenomena such as aging can be averted by the utilization of CRISPR. Senescence is the process of the physical body deteriorating due to a numerous number of causes, the most prominent causation being that the senescent cells accumulate. The buildup of senescent cells are the direct cause of the formation of wrinkles. Senescent cells are caused by the failure of DNA, or more specifically, the chromosomes. When a cell is conducting mitosis, the cautiously labeled chromosomes are being unlabeled in order to replicate the genes as well. While conducting this process, a microscopic segment of DNA is lost from the gene pool forever. To prevent the important parts of the DNA from being gone, repeated nucleotides, referred to as telomeres, are constructed at the ends of chromosomes that act as the hard ends of shoelaces. Unfortunately, with each cellular division, the telomeres shrink with each division. Ultimately, the telomeres will become used up and the cell will become senescent. This is a major problem for organisms with long lifespans, as the severe buildup of senescent cells will disable the function of organs. For instance, due to senescence, there is a heightened risk of kidney failure or liver malfunction, and a greater probability of being diagnosed with a form of cancer. In an experiment, mice were genetically enhanced such that the senescent cells diminished in building up. The case study was a triumph for that scenario, but it is safe to assume that engineering every single one of the 70 trillion human cells will not be a viable idea. Fortunately, there is another solution to the crisis. Scientists observed that a peptide called FOXO4 was critical to whether or not the cell could self-destruct when the cellular processes were failing. Hence, by injecting FOXO4 into senescent cells, they could revive the cell and solve the problem of senescence. Cancer can also be solved by genetic engineering. Cancer is caused by cancer cells, which have formed due to cellular failures and mutations. Cancer, in essence, resembles another entity stored within our body, fighting for the same scarce nutrients and attempting to dominate over the other. Despite containing an uncountable number of cells, our body is incredibly gullible, particularly the immune system. With the correct mutation, cancer cells can become shredded from detection and hence become almost indestructible. CRISPR, however, can change that. By editing the very genetic sequence of these mutational cells, the cells will re-enter the cell cycle, and hence the cancer cells can become a healthy cell once more. Thus, the artificial identification of DNA proves to be beneficial in some aspects. Out of a nutshell, what do astounding attractivity, aesthetic and airy voice, and 5 rows of teeth contain in common? It is evident that these 3 elements are all interconnected by the formation of DNA, and hence are related to life. Artificially altering our genetic sequence contains the potential to remove common hereditary diseases from genes, but once it falls into the wrong hands, also contains the potential to alter the definition of life. In a nutshell, life is constructed out of a randomly intertwined chain of nitrogenous bases referred to as a nucleic acid, called deoxyribonucleic acid, or DNA for short. DNA codes for everything pertaining to life, from the hues of our pupils to the number of synapses in our brains to the success probability of mitosis. DNA can exist without life, but life cannot exist without it. Bacteria utilize an enzyme referred to as Cas9 that contain the potential to scramble DNA, and therefore alter the sequence. Humans usurped this tool and used it for our own selfish purposes, and dubbed it as CRISPR. The reconstruction of our genes can be fathomed with a simple scenario. Imagine that you are driving a rundown car on a dilapidated stretch of highway. You are aspiring to build a 5-star Mercedes van with nothing but road junk that you find discarded with the rest of the junk on the road. Gene engineering is incredibly fluctuating and volatile. The consequences of engineering our existence are still to be ascertained, but all we have to do to navigate the junk-riddled, litter-strewn path ahead is to believe in the possible. This special video is accompanied by a website, which we provide various things, including an interactive infographic that models the system of genetic engineering, a detailed definition of DNA and CRISPR, a graph, and so many more things. Go check it out in the video description below. This was the final video for 2022, and it has been a truly interesting year. We have started the channel merely in May, and we have already come so far. Our animations have evolved, while the digital layout of our website enhanced exponentially, as well as being translated into 5 additional languages. We cannot wait for what's up the road. We look forward to seeing you next year. Happy holidays.